This is Ashutosh Yadav and welcome to my YouTube channel AkyBiotech. Friend, as I promised, this video is the part 2 of the memory based question paper of ICAR-ASRB-NET in agriculture biotechnology. Hope you like the part 1 video. Part 1 video have the 60 memory based question paper, and this part 2 video have the 50 memory based question paper of ICAR-ASRB-NET in agriculture biotechnology. Hope you like this. Friends, before starting this video, if you are new at my channel, please subscribe the channel, like and share the video.
Now let's take a quick revision of the next 50 questions. Number 1. Which enzymes are used for scavenging reactive oxygen species in cells? Answer is SOD, catalase, ascorbate and tocopherol, etc. Number 2 question. Dark reactions take place only in dark condition. True, false? Answer is false. Number 3. Cry1AC is specific for which group of insects? Insect group is Lepidoptera. Number 4. Topoisomerase is a form of gyrase. Next question. If the 2n of corn is 20, then what will be the chromosome number in endosperm? Answer is 30. Number 6. Transposons were discovered for the first time in maize by. It was discovered by Barbara McClintock. Number 7. Isolation of promoter is possible through which approach? Promoter trap is the approach used for the isolation of promoter. Number 8. Sticky end in DNA fragments is made up of few unpaired bases. Number 9 question. What is the insert size that can be cloned in back vector? 150 to 300 kb is the answer. Number 10. The gene sequences found in different species of organisms but performing same function is known as. They are known as orthologs. Number 11. Name the vector which can accommodate largest insert size. Yak. Next. Number of different enzymes involved in glycolysis reaction pathway. 10 enzymes are involved in this pathway. Number 13. Plasmid doesn't contain centromere, antibiotic resistant gene, origin of replication. Answer is centromere. Name the smallest binary vector. P green is the vector. The natural function of restriction enzyme in bacteria is to digest the foreign DNA. Number 16. An embolic process is generally performed by which operon? It's the repressible operon. Number 17. What is the role of cyanide in oxidative phosphorylation? To inhibit cytochrome oxidase. Number 18. AFLP is performed on which gel? It's performed on 10% page. Number 19. Which group of enzymes is involved in cleaving gene? Nucleases. Number 20. Cold responsive in plant is developed by which of the following strategy? The strategy followed is engineering the gene producing antifreeze protein. Number 21. What is false about SNP? Is it a dominant or a codominant marker? So, it's a codominant marker, hence the false statement is it is dominant marker. Number 22. Which technique is not tagged as next generation sequencing? Sanger sequencing. Number 23. Which organelles are absent in plant? Lysosome and centrioles, as we all know, are in animals. Which is the most widely used method for gene transfer in plants? It's agrobacterium mediated transformation. Which is an osmoprotectant? Glycine betaine. The first discovered molecular marker is RFLP. 1 picogram DNA contains how many base pair DNA? It's 970 megabase pair. BLAST N refers to? It refers to searching for the nucleotide query sequence against a nucleotide sequence. Number 29. When one enzyme has 2 substrate binding sites, then the enzyme kinetics and binding affinity is represented by which curve? Scat chart plot. Number 30. Carbon dioxide in plants is fixed by which process? Calvin cycle. It's called the dark reaction of photosynthesis. Next question. Higher COT value indicates which type of DNA sequence? Unique sequence. Anticodon on tRNA for AUG on mRNA is? Answer is UAC. Softening of tomato is mediated by which enzyme? It's mediated by polygalacturonase. Glucose is utilized first when other sugars are also present by which mechanism? Catabolic repression. Which compound works against oxen? TIBA. 3D structure of protein can be determined by which method? X-ray crystallography, NMR, etc. Amino acid most frequently present in a helix of protein is aniline. Write the name of one gene prediction software. GeneScan. Herbicide phosphinothrycin is inhibited by which enzyme? Phosphinothrycin acetyltransferase, PAT. Apoenzyme and coenzyme are together called as, they are known as holoenzyme. First somatic hybrid was developed by Carlson et al. The first discovered molecular marker is RFLP. Extranuclear DNA is found in which organelles? It's mitochondria and chloroplast. What are ribozymes? RNA with enzymatic activity. Now comes the next question. Which is not a part of promoter? It's terminator. 45 number. Which is not an aromatic amino acid? Histidine. The innermost membrane of cell which surrounds vacuole is known as tonoplast. Which type of hybrid forms in northern hybridization? It's DNA RNA hybrid formation. Next question. Antibody protein reaction takes place in which type of blotting? It's Western blotting. 49 number. Which is not determined by northern hybridization? Protein sequence is not determined by this. 50. Promoters are cis-trans acting elements. As we all know, they are the cis acting elements.